This is a production of Cornell University.
And welcome everybody to the Fall 2020 seminar series online. This is really exciting. I'm particularly excited for our seminar speaker to kick off with apples as we get into the apple harvest. And today's seminar speaker is Myles from Dalhousie University in Nova Scotia. I first learned about Sean from his research in apple genetics, and while I was working on the horticultural side, he's been working a lot on the genetic side. We have a lot of overlapping interests, and that really became apparent in our conversation just before this seminar today. But in doing a little bit of research on Sean, it's pretty interesting, and it's always really fascinating to learn about people's academic histories. And really, Sean's training was in human genetics, and he was studying the populations of different people around the world. He was working at the Max Planck Institute in Germany, and actually was working on some of the earliest fast sequencing machines, and being able to process through many samples and doing things like SNP arrays for humans to understand how different populations and people moved around the world. And then got into grapes basically because he fell in love with a winemaker and figured that if they're going to find a place to live together, he better figure out something that he can do where she's going to be based, because grapes can only be grown in so many locations throughout the world. And got into apples later in his academic interests. So really fascinating and deep research history that Sean has. As we do for all of our seminar speakers, one interesting fact, and even though all of these things I mentioned already were quite interesting, I asked Sean if there was something he'd like to share. And so the interesting and not well-known fact about Sean Myles is that he can tap dance. And so you can think about him tap dancing while he does his seminar today. So I'll pass it off to Sean at this point. Again, if anybody has questions for Sean during the seminar, feel free to put them in the chat and we'll address them during the Q&A period.
Great, thanks, Greg. I'm just, if anyone has any problems hearing me, then they can put something into the chat there to let them know. Thanks for the introduction. I was just thinking, you know what, it actually would make the seminar a lot more interesting if I was tap dancing while doing a talk about genetics. But I would need a little more practice, I think. So I didn't really quite plan on that, but I'll keep it in mind for next time. So I'll expect another invitation. Thanks for having me. This is really great. And I wanted to mention too, I'm going to share my screen here, I think, right? I think this is how this works. So that if I pop this up, I believe everybody should be able to see that. Great. So I did want to mention, you know, you talked about this sort of circuitous route towards agriculture, and it's worth, since there are many students listening in, I hope that, you know, often it's a little bit daunting when showing up somewhere and getting into something new, and when you're getting into a new subject area. And I want to assure you, you know, I did my undergraduate degree with a major in English literature and my master's and PhD in human genetics, and didn't start looking at plants until my postdoc. And so, you know, these sorts of circuitous routes can happen in life, and there's lots of chance meetings along the way. If you put your head down and focus on doing good science and doing good work, then lots of things are possible. And so I thought I'd mention that, because I know that often, in fact, every time I took a new position somewhere, at each institution that I went to, I would feel completely overwhelmed when I first got there, like I did during my postdoc at Cornell. I arrived in Ed Buckler's lab, who was talking a million miles a minute about corn, and I knew nothing about anything anyone was talking about in lab meetings as a postdoc. You know, you kind of feel like you should probably know something. So that feeling of being overwhelmed and being stressed out and like feeling like you're an imposter and you're in the wrong place, I can assure you that it does subside with the years, and that's my little piece of advice for all the students getting in and shuffling around into their different departments and different endeavors along their career paths. So thank you very much for having me. The first thing I'd like to start with is that to show you where I am. So I'm in Nova Scotia, Canada, up here in the sort of northeast of the North American continent, out on that little peninsula, and it's a beautiful place to be. And I thought I'd just describe a little bit where I am here. This is Nova Scotia. There's nothing of the US on here. Maine is just west of New Brunswick here. But we are in, I'm located in the Annapolis Valley. I work for Dalhousie University, but I actually work out of a federal research station. So it's kind of like Cornell professors who are co-located up in Geneva, New York. I actually work out of a federal research station here in Kentville, Nova Scotia, up here in what we call the Annapolis Valley. And the Annapolis Valley contains about, we grow about 10% of Canada's apple production. So it's a very small area, but it's sizable in terms of its contribution to apples in Canada. And there's only about 1 million people who live in Nova Scotia overall. So it's mostly rural, and the main city is Halifax. We grow in our little valley here, grow almost, you know, the majority of the food grown in Nova Scotia, especially all the fruit crops. So we grow apples and pears and plums and peaches and more grapes. There's a growing wine industry. So it's not unlike upstate New York in its growing conditions and the sorts of things that we're growing. And it's a beautiful place to visit. And when all this COVID stuff is over, you're more than welcome to come and have a glance. We don't often get visitors coming up to talk about science, so it's a real pleasure if you do. So if you were ever thinking of having a visit up to the east coast of Canada, please do get in touch, and I'd be happy to show you around. And it's a beautiful place to come and eat apples and drink cider too, of course. So I did want to mention, you know, during my postdoc when I was at Cornell, there was a real interest in using modern genomics technologies and the emerging genomics technologies to characterize the USDA's gene banks or germplasm collections. And my postdoc project under the supervision of Ed Buckler was to develop a SNP array and characterize the USDA grape germplasm collection in collaboration with the USDA researchers up in Geneva, New York, and also part of the collection is down in Davis, California. And it was a wonderful opportunity. I felt really fortunate to be able to do that work. And I've moved on now, started a position up here in Canada. And in our experience dealing with the USDA germplasm collection, of course, is that it was not planted for the purposes of doing genetic mapping work necessarily. It's planted as a gene bank in order to preserve and maintain diversity. But of course, that often means that the plants are planted at different times, perhaps on different rootstocks, maintained by different people, and the data is collected at different times of the year and in a different way than you might want to collect the data for the purposes of genetic mapping. And so when I started my position here in Canada, I made a special request to the USDA that was accepted, fortunately, to actually make a copy, essentially, of the USDA apple germplasm collection up in Nova Scotia, which we now call Canada's Apple Biodiversity Collection. It contains as many of the Malus domestica, so the domesticated apple, and its primary wild ancestor, Malus sieversii. And so we didn't take all the other wild species and incorporate it in here, because the purpose of this population was really to plant it out specifically for the purposes of genetic mapping and doing genome-wide association studies. So we planted this out, each of these varieties out in duplicate. Well, we grafted them all to M9 rootstock in 2011. And then in 2013, we planted it. And we ended up with 1,113 different accessions planted in duplicate with check varieties that are planted throughout so that you can control for the spatial effects in the orchard. And so in total, it's 2,354 trees. And that includes, you know, most of that material is from the USDA, but there's also advanced breeding lines from Canada's apple breeding program and some other stuff we had hanging around in Nova Scotia that we threw in there. So if you're ever interested in seeing this, so what we've been doing, to give you an idea, I'm not going to talk about this collection today. This is really the genetic mapping work. I'm going to talk more about the evolutionary work that we've done based on the USDA collection. But what we've really focused on here in Nova Scotia with this apple biodiversity collection is to try to get as much phenotype data as we can on a budget, of course, And it's very time consuming and expensive to do, as many of you are aware, to collect traits across an orchard like this. And so we've made use of barcode scanners where we can to speed that up. And it's a Herculean effort really to do over 1,000 varieties and harvest them all at the right times too, because they're all developing at different rates, and put them through, you know, and collect the data that you'd like. So I made a little video here, and I'm hoping this is going to work, and we'll see. But this is one of the devices we use called the Pimpernel that does automated weighing of every apple. And then so the first step is the weighing right there. And then the next step, it goes up into a penetrometer. There's an automated penetrometer that will take a firmness measure. Then it shoots that apple into a chute that then gets squished, and the apple gets squished up, and it does automatically. It takes the acidity and the soluble solids, and so you get that automatically directly into the spreadsheet, no writing down data and copying it back into Excel files and so on. And we made that, trying to find ways of making that data acquisition process as efficient as possible. And one of the other little devices is pretty nifty because we want to get color and shape. We take a picture of all the apples going through. You call this little device the Snapple because it takes snapshots of every apple. And you go and put the apple in, and it takes a snapshot and links it to the ID, and it goes right up into a little drive there. We put this together with some engineering students at the university. And so that sped things up, and we've got a decent data set from our apple biodiversity collection. And since many of those are actually apples that are found at the USDA collection in Geneva, that the data set that we will come to publish here shortly will be, you know, it'll be publicly available and available to anyone who's also studying these apples in Geneva, New York. So I encourage anybody who is doing work in apples and is looking for sort of a comprehensive evaluation of phenotype data that they might like to link to the data that they've got. And the genotype data is going to be all available as well. I think we've got about 250,000 markers now genotyped in the entire collection. So if you'd, we're really keen to know of other people who are doing this kind of work on those same varieties, and since they're genetically identical but just planted in different spots. If you're up in the Geneva collection doing something interesting, then feel free to reach out, and we're happy to come up with a way of helping out your study and perhaps adding some data to it that will be useful to you. Okay, so today I'm going to talk about some data that we've collected on the USDA collection, which is far overdue to be published, but it should come out relatively soon. This is work that was led by two postdocs in my lab, Zoe Mijakovsky and Kyle Gardner, and some help from a PhD student, Sophie Watts, and my main collaborator, Jun Sung from Agriculture and Agri-Food Canada here, who helped out. And this was a project that was led in collaboration with the USDA, led by Ganyan Song in Geneva. So the USDA apple germplasm collection is fantastic. It's really a real showcase gene bank from the USDA in terms of its breadth and depth. Certainly, the number of countries represented in terms of where these apples are coming from is enormous. You can see a lot of them are from the United States, 228, but you know, there's 100 varieties from the United Kingdom, and a good representation from Central Asia, which is sort of the original home of the apples, or where it originated, so those progenitor apples. And so this gives you an idea of the breadth of that collection. And what we did is we collected genotyping by sequencing data. I'm going to talk about some genetic data that we collected from this entire collection. We ran 42 lanes of Illumina HiSeq, so generated 7.8 to 7.9 billion 100 base pair reads from 1,949 accessions from that collection. I'm not going to talk today about what we did with the other members of the Myles species that have not really contributed, or contributed only recently, really, to apple breeding and apple development to modern cultivars. I'm focusing mainly today, exclusively today, on the variety, on the species thought to have contributed to modern day apples. So we've used these data already, actually, in a previous publication to do a genome-wide association study to perform some genetic mapping, where we take the data from USDA GRIN database. And so we didn't collect any phenotype data ourselves. We just used existing historical phenotype data from the database and linked it to the GBS data, the genotyping by sequencing data. And that resulted in about, at that time, when we were doing fairly simple bioinformatics with these data, we had 8,000 SNPs, or single nucleotide polymorphisms. These are the markers that we get from the sequence data. So we only ended up with 8,000 SNPs and some very messy phenotype data, I must admit, from the USDA GRIN database. Often things are scored as, you know, the apple is either soft or it's hard, but not a quantification of the actual firmness of the apple, for instance, or the apple is either early or it's late, but not the actual date it was harvested on, and so on. But despite that, you know, even with some relatively noisy phenotype data, you can catch the lowest hanging fruit. And we did in that publication demonstrate that we identified the main locus underlying firmness that we've done some subsequent work on, and believe that it is very important for ripening, apple ripening. So I'm not going to talk about that work today, but I do want to point out that it's really great using these genotyping by sequencing data, you know, or any of this next generation DNA sequence data compared to microarrays. When you're using a microarray, you run it once and the data you've got is what you've got. But it turns out with sequence data, there's always a new algorithm coming out, you know, every year to be able to point at your original sequence data to extract more of that data. Right? So you can, we spent a couple of years developing imputation algorithms and genotype calling algorithms that would be more suitable for non-model organisms like the apple that have relatively poor reference genome, where the order of the markers is not always really reliable, where you can't really phase haplotypes because of rapid linkage disequilibrium decay, and where the existing genome sequences are kind of a poor quality, and they're highly heterozygous. So those present some challenges for genotype calling and generating a really good genetic data set. And so we worked quite hard on this and developed our most recent imputation algorithm, link imputer. And with link imputer, we've actually managed to generate a lot more SNPs in the present presentation that I'm doing today, and the paper that we've got submitted. We've got about 20 to 30,000 SNPs. So you more than double the number of SNPs you got from your first try. But since then, we've actually tweaked things a little bit, and we're up over 200,000 SNPs now, with the exact same data that we used to only get 8,000 SNPs from. So again, with microarrays, you get what you get. But with the sequence data, there are ways of using that missing data that's in there, using patterns from the existing data to fill in missing data quite a bit better, we find. And that's been a real benefit to us. So just keep that in mind, in that link imputer algorithm for people who are working on non-model organisms and collecting genetic data, and they wish to fill in their missing data. Link imputer is great because it allows you to say, for example, you know, I'm okay with a 10% genotyping error rate if I can get 250,000 SNPs. Or you can say, no, I want really high quality. I only want 1% error rate. Well, then you might only get 25,000 SNPs. And it allows the user to decide what error rate they wish to accept, and therefore how many markers you're willing to get. So if you're on a fishing expedition and you're doing some genetic mapping, you may want to make it pretty loose and have large numbers of markers with, you know, quite a bit of error, because you can always go back to the markers that are associated and go check to see whether they're real. On the other hand, if you're doing some kind of population genetic analysis and you want reliable marker data, you may want to be more strict. So it allows the user to decide that. Okay, so I'm just going to get into some of the questions that we wish to answer with these genetic data, and it's well known from several publications and from lots of work that's even been done on the phenotype level and just traditional botany, that the primary progenitor of the modern apple that you would see in the grocery store comes from a species called Myles seversii, which is still found in the forests of Kazakhstan in the Tian Shan Mountains along the very close to the border of China. And so here you see Myles seversii here printed right in the middle of Kazakhstan, and that it's believed that this Myles seversii then, as humans moved along with humans over to Europe, and there it had some introgression from Malus sylvestris, which is another Malus species. It's a wild European crabapple, very small, astringent, and bitter. And the degree of that introgression is still under debate, and there's been some conflicting findings in the literature. So one of the things that we can do with our data, since we have some Malus sylvestris in the collection and some Myles seversii and lots of modern apples, we can ask ourselves the question of what do the relationships look like between these various species. So here are the sample sizes across the top for this particular analysis of 749 Malus domestica. So those are what you would consider dessert apples and cider apples, modern varieties, and so on. 115 Malus sieversii, those are from Kazakhstan, and 11 Malus sylvestris from Europe. And so you can see if we run a principal components analysis, if we take those 20 to 30,000 SNPs and we plug them into principal components, what principal components allows us to do is visualize the relationships among all the individuals in 2 dimensions. When you have 20 to 30,000 SNPs and you want to visualize how everyone is related, essentially PC1 here is like shooting a regression line through a 20 to 30,000 dimensional space. And then PC2 is then another orthogonal kind of regression line through that space. And so it's capturing as much of the variation in the data as you can. And you can see PC1 accounts for about 6.5% of the data, 6.74% of the data. So what you're looking at here gives you a visualization of the relationships among individuals. And what you see, the squares on the right-hand side of the plot, those are all Malus sylvestris. Those are the European wild crabapple on the right-hand side. You can see that there's 2 groups of them. And in fact, the one in the top, they're from Germany, and the one down below are from further in Eastern Europe. So there's a little bit of structure within the population, but we're really just concerned with PC1 here and how very easily the genetic data separate those Malus sylvestris samples represented by the squares on the right with the triangles on the left-hand side, which belong to Malus sieversii. So it's very easy with these genetic data to pull these two species apart. Now, if we want to know where the Malus domestica lie and how they're related to these 2 groups, then what we do is we project those Malus domestica onto these axes. And there's a way of doing this using standard principal components analysis to project those samples on. And I've colored those dots, either they're gray if we don't know exactly what they're used for, or if they're cider apples, they're in orange. And if they're dessert, mainly what we call dessert apples, or the ones eating apples, the ones you find in the grocery store, or other, some of them are labeled as cooking in green, but they're essentially not cider apples. They're in blue. And so you can see that basically you form a cloud right in the middle, right between Malus sieversii and sylvestris, about 50-50. So the conclusion there would be that they share about half their ancestry from the original ancestor Malus sieversii and about half from Malus sylvestris. However, I'd be cautious about putting too much into that interpretation, given the number of markers we have and the number of Malus sylvestris samples we have, which is only 11. And there are some more studies coming out that we know of here in the near future that are doing whole genome sequencing, which will be able to better answer those questions. But despite that, one of the most interesting things here is that those 2 clouds of orange and blue dots don't actually line up completely. The orange dots tend to be pulled over further to the right, and the blue dots further to the left. And that's demonstrated here in panel B, where if you look at the PC1 values of cider apples, you can see that they are significantly greater than the PC1 values of the other or dessert apples. And so it appears from our analysis that regardless of what the proportions are of the ancestry between sylvestris and sieversii, that cider apples tend to, they appear to derive more of their ancestry from Malus sylvestris, the European crabapple, than do dessert apples. And this makes good sense in a way because the Malus sylvestris apples are known to be very astringent and bitter and have a lot of tannin and are acidic and share some of those characteristics that cider apples, that are desirable in cider apples and that are less desirable in an eating apple. And so it may be the case that some of those desirable characteristics from cider apples are actually introgression events from Malus sylvestris. We weren't able really to do a piece-by-piece genome analysis to see where those introgressions might occur given the SNP density that we have, but I'm sure that's coming in the future too with future data. Another question we can ask is what regions of the genome appear to have been under selection during domestication? So what are the regions of the genome that really are most differentiated between Malus domestica, or the apples that you might see on the grocery store shelf, versus the wild ancestors that are growing in the forests of Kazakhstan? And to do this, what you can do is calculate FST, which is a measure of allele frequency difference between the 2 populations, between Malus domestica and Malus sieversii. And FST is a very simple measure. If the allele frequency at a particular SNP is exactly the same between the 2 populations, you get an FST of 0. if it's a fixed difference, if all the domestica are homozygous AA and all the at that locus and all the Myles sieversii are homozygous TT for the other allele, then that's a fixed difference, then they'd have an FST of 1. You can see that there are regions of the genome that are approaching an FST of 1. Each of the dots on this plot represents one of the SNPs that we've genotyped. And the horizontal red line represents the 5% threshold genome-wide. So any dots above that line are within the top 5% most extreme FST values in the genome. And what we expect when selection happens at a particular locus is the markers there, they'll be driven to high frequency very, very quickly over time, and there'll be a large FST value between domestica and sieversii. So we're taking a look at all those dots across the top and thinking, okay, are those lying in any genes that are particularly important? Well, there's a somewhat orthogonal analysis you can do here to determine whether it's really selection or not, because you can make an FST distribution from two populations when no selection has happened, and there's still going to be 5% of your markers that are going to be within the top 5%. There's always a top 5% to every distribution. So we throw another analysis at this called XPEH. So when a marker rises in frequency very quickly due to selection instead of just genetic drift, then what happens is recombination doesn't have enough time to go and break down the haplotype that's around it. And what you end up with is a very long haplotype that's homozygous in the population. And that can be calculated as this statistic called cross-population extended haplotype homozygosity, or the XPEH. And that's shown there on the y-axis on this bottom panel. And each of the dots again here represent one marker that we've genotyped across the genome. And its position along that axis shows whether it shows extended haplotype homozygosity in Myles domestica. So here we're checking to look for evidence of selection within the domesticated apple. So dots that lie above that horizontal red line represent potential targets of selection during domestication and breeding. Now, ideally in these circumstances, you see a great big peak that overlaps with FST and with XPEH, and right underneath that peak, you look into the genome browser and you see the name of a gene that is really obvious, and then you go and you look at it in detail and you characterize it in a tomato and you throw it at Nature Genetics and you get published. Well, it doesn't turn out that way for us, I'm afraid. With no smoking guns, I mean, you look at the gene list and it's lots of different genes. And so one of the things you can do when you don't have any real great candidate genes for domestication between the domesticated apple and its ancestor, you know, it's worth keeping in mind that when we look at the ancestor of the domesticated apple, they often really look the same too. Like they look very similar. Sieversii apples sometimes look like something you could find in the grocery store. Whereas in other animals and other plants, when you look at the wild ancestor and you look at the modern cultivar, it's really obvious. It's like, oh, okay, it's seed shattering, or it's, you know, ear development, or it's something. And in apple, it's really not entirely evident what genes we would look at if we were going to look at something that's been under selection. So we don't have really good a priori ideas about what they should be. But so one of the things you can do, you can take a look at all of the SNPs that overlap within the top 5% for both FST and XPEH. Those represent potential targets of selection, and you can look at whether genes that are within a certain distance, I think 50 kb we chose, and then you can see whether those genes are enriched in any particular biological category. Are they involved? Are they enriched in some function? And when we do a gene ontology enrichment analysis, we do find these three things that are enriched for these genes that appear to have been, that show signatures of selection in Myles domestica. And that's target of rapamycin signaling, cellular response to phosphate starvation, and cellular response to stress. It's been suggested before too in a previous publication that the targets of selection in the domesticated apple are related to adaptation to new environments as they're moved from the Tian Shan Mountains to new environments. And our data are consistent with that hypothesis, but we don't have any really great smoking guns and single genes that we have a great story about right now in terms of domestication in apple. But we can do the exact same analysis actually by comparing dessert apples with cider apples. So there are 69 apples in the collection for which we have genotype data and good phenotype data that are characterized as cider apples. And these are, so in some cases they're characterized as both. These are the ones that are characterized as just cider apples. And dessert apples, we have 288. Here's a comparison comparing FST between dessert and cider apples. And you can see the FST values are generally lower. They don't go all the way up to 1. The y-axis there is only going up to 0.5. So they're more closely related to each other than Malus domestica and Malus sieversii. That makes good sense. And what we see here is that the same kind of analysis can be done, but in this case, we can look at genes that appear to have been under selection according to this XPEH statistic in dessert apples. Those would be found above the dotted red line here in below. And then those that appear to carry a signature of selection in cider apples. So what may have been selected for in cider apple development and cider apple breeding. And again, we don't find any single genes that stand out as being the sort of smoking gun in this analysis, but when we do a gene ontology enrichment analysis, we do find that flavonoid biosynthesis genes are enriched in the regions of high FST and high XPEH in the dessert apples, while malate transport is enriched for cider apples. Now, we know that cider apples in general are more acidic, and malate transport genes are involved in the production of malic acid. So it's very possible that this is something that's been selected for in cider apples. And flavonoid biosynthesis, flavonoids are molecules that contribute to the tannin and the mouthfeel and the polyphenols in an apple and a lot of the Greg can help me on all this stuff. I think he knows a lot more about this than I do. But the flavonoid biosynthesis in dessert apples, one might think, well, I don't think, in terms of the difference between dessert and cider apples probably have fewer polyphenols than cider apples. And it may be exactly that. It may be that there's been active selection against polyphenols and antioxidants. And in fact, we have some orthogonal data here to speak to this. This is the phenolic content in apple varieties compared to their year of release. So these are all dessert apples that have a year of release associated with them. And you can see that it's the only thing we've found actually that's correlated with year of release. We've looked at soluble solids and size and acid and everything. And the only thing that seems to be really robustly correlated with year of release, this is the year that the apple cultivar was released to the public as, after breeding, is phenolic content. Seems to have gone down. You can see that in the year 2000, the average for apples that were released after the year 2000, their phenolic content is on average, I think, something like 30% lower than things that were released before 1900. So it does suggest that perhaps there has been active selection by breeders for reduced polyphenol content in dessert apples, making them less astringent, less bitter, and less tannic, and that kind of thing. So this is worthy of future investigation. But undoubtedly, the strongest signature of selection that we find in the genome is for color. So there's, on the top panel here, you see the result of a GWAS for color with these data. So in GWAS, we take a look at every marker and it's the strength of its correlation with a trait. And in this case, the apples are characterized in the USDA GRIN database as either red or green, or red or not red, I think. And so we've actually just done an analysis here of red versus green apples. And when we compare red versus green apples and we calculate and we do a GWAS where we're looking for the correlation between each marker and that color, the strength of the correlation is indicated by the height of the dot on that y-axis. So that y-axis is the minus log 10 p-value of the strength of the association. And again, that horizontal red line that you see across here, that represents our significance threshold. So dots that are above that, we're going to consider significant in this analysis after correcting for multiple comparisons. So you can see that there's one very strong peak here on chromosome 9, and that's unsurprising. It overlaps with the red TE, this transposable element that we believe is responsible for the red color in apples that's been described in another manuscript here. And it sits in the middle of a MIB transcription factor, which is known to be involved in anthocyanin synthesis. So this makes good sense. We're picking up the locus using GWAS. This isn't surprising. There are some little helicopter, what we call helicopter hits, flying around here on the outside. Turns out those actually are not other loci involved in color, but they're mismapped SNPs. They actually belong on chromosome 9, but the genome is often wrong. And so you end up with these little dots around the genome and they're just in the wrong place. So we really have one single locus, a very strong major effect locus controlling color. And if we look at the XPEH again and we compare red to green apples, it turns out that red apples have a very long extended haplotype around there, and they're often homozygous for this haplotype. So we see this extended haplotype homozygosity, this very strong signature of selection around that MIB locus, suggesting that breeders over the last, we can't really say how many years, but certainly over the last several hundred years have been selecting for redder and redder apples. We can do, similarly, you can think of other phenotypes that may have been desirable during apple improvement and that may have been targeted and that we may be able to find signatures for in the genome. And one of those might be firmness. Now here we're presenting just firmness at harvest. And I understand, you know, firmness at harvest is one thing, but firmness after a few months of storage is a completely other thing. So how much do you soften? So I'm not looking at softening here. I'm really just looking at firmness. And so firmness at harvest, if you run this in the USDA GRIN database, it's characterized as either firm or soft. So we take those two and we run a GWAS with our markers. This is with 8,000 markers in a previous publication. And we find this, again, this is each marker and its strength of its correlation. The higher on the y-axis, the stronger the correlation. And the dotted line here is the significance threshold. And we find this one hit on chromosome 3, and it falls within NAC 18.1. And NAC is a transcription factor that's known to be involved in the ripening process of lots of organisms. In fact, its homologue in tomato is the NOR gene, and the NOR mutant in tomato is a well-known mutant, which is a knockout of the NOR gene that inhibits ripening in the tomato. And so, and in other organisms, we know that NAC genes are involved in ripening. So this is an excellent candidate gene. This is one of these cases where we did have a smoking gun. You know, we do the analysis, we find the gene, it looks like NAC 18.1. We've done it again many times. It's been verified in other collections, but certainly when we do it in our whole apple biodiversity collection with 863 varieties, you can see that it comes out, that same SNP actually comes out as the top SNP. You can see how much more power you have when you measure firmness here using a digital penetrometer and measure the actual firmness and get a quantitative measure. And you have, you know, 4 times the sample size or twice the sample size maybe. So you can see that NAC really comes out very, very strong here. The question is there any signature of selection around that locus? Is there any evidence that there's been selection for firmness during apple improvement? And the question is, well, kind of maybe. So the firm versus soft apples that we do with our 20 to 30,000 SNPs in this particular paper that I'm presenting here, in this analysis, we see that NAC 18.1, that green dot again, that's the marker in the middle of NAC 18.1. You can see it does not go above the line and is not significant in this analysis, okay? Despite the fact that we know now that we've done this in other populations, and the folks in Denmark did it, an analysis, and they also found NAC 18.1 associated with it. Another European group has. We know that that's really a SNP that should be associated significantly with firmness. But this is a good lesson for people when they're doing genetic mapping, is like, where did that horizontal red line come from? Who decided that it was going to be there? The fact of the matter is in this analysis, we have 20 to 30,000 SNPs. So when we correct for multiple comparisons, that red line goes up. So the criteria become more stringent. If you have fewer markers, the red line goes down. But if NAC 18.1 is part of the marker set, in one analysis, it's significant. You add markers to your analysis, all of a sudden it's not significant anymore. So it doesn't mean significance, statistical significance with these somewhat arbitrary thresholds. It's very different from biological significance. It's worth keeping in mind that just because NAC 18.1 falls below the horizontal line in this analysis doesn't mean that it's not biologically relevant, because we have evidence from other studies that it is. So where does it fall in terms of its signature of selection, in terms of its haplotype homozygosity? You can see here on the bottom panel, that green dot is the SNP that lies within NAC 18.1, and it is not within the top 5% of SNPs genome-wide, but it's within the top 5.2% of SNPs genome-wide. So you're in one of these situations again where it sits right around this threshold of significance that you've made for yourself. And so the evidence is a little bit ambivalent. So it's worth pointing these things out, because sometimes you wish the analyses would come out and provide you smoking guns every time, but it doesn't always work that way. So we do find some evidence for selection for firmness, but certainly future studies are going to be required in order to validate that. So perhaps one of the most interesting things that you can do, is take the data and actually calculate how closely related are all the samples to each other. So make a giant pairwise genetic relatedness matrix, and then ask yourself, okay, how many of these samples from the USDA collection are clonally related? They're actually essentially genetically identical, barring some genotyping error. You can see most accessions in the collection do not have another clone in the collection, but about a quarter of them do. They have at least one other tree in the collection that is named differently that is a clone of it. Okay? And some of them have multiple trees that are all actually the same variety. So in the case of Golden Delicious, there are 22 Golden Delicious in the collection. There are 15 Red Delicious, 10 Macintosh, and 8 Northern Spy, right? And you can represent that here as a network where every single dot here is an accession in the collection, and a line between them, or an edge in the network as we call it, represents a clonal relationship. So you can see that there are quite a few that have one clonal relationship. 2, 3, 4, 5, 6 isn't very common, but then 7 or more is really quite common. So we see several varieties that have 7 or more, and those are the popular varieties that have been propagated out and where multiple clones exist and are used. So you can do the exact same thing. Instead of looking at clonal relationships between varieties, you can look at first degree relationships, so either sibling or parent offspring. So again, here in panel C, on the left-hand side here, you see each one of these nodes in the network represents an accession in the USDA apple germplasm collection, and an edge or a line between them represents a first degree relative, so either parent, offspring, or sibling. So you can see we have in this histogram on the right-hand side, most varieties have 0. Like there are many, many cases where a variety does not have a relative in the collection. But then you see many of them have 1, a little bit fewer have 2 first degree relatives, 3, 4, 5. But some of them have more than 7. Golden Delicious and Red Delicious have over 60 first degree relatives in the collection. So when we talk about, and 15% of the USDA collection are actually related to the top 8 varieties that are grown within the United States right now. So you've got a huge amount of relatedness inside this. Over half of the collection actually is interrelated in a series of first degree relationships in this network. You can see how there's a big clump there that is all sort of interconnected. That makes up over half the collection. So when we say that ultimately when we want to do genetic mapping and we want to do it in a large population of unrelated individuals, well, this is not going to happen in the apple. Like all of these apples, they're like a big extended pedigree that you've got inside that collection. And you can quantify the degree to which they're related in first degree relatives in this manner. What's particularly interesting, I find, is that when you look at those first degree relatives and you highlight, among the varieties that we've genotyped, we have, out of the top 10, we have 8 of the top 10 cultivars that are grown in terms of acreage in the USA. So, or according to the apple grower website, something or other. You got to pick some sort of source. But anyway, these are top, like 8 very well-known, widely grown varieties in the United States are highlighted in red, those nodes. And you can see the, again, first degree relatives are all connected with those edges in the network. And you can see that there are numerous first degree relationships, Golden Delicious and Red Delicious, again, having over 60 first degree relatives. And that 15% of the USDA collection overall is actually connected by a first degree relative to these top 8. And that the top 8 in each other, they're related to each other through a series of first degree relationships. So this is not all that different from what we found in grape as well, where Pinot Noir and Chardonnay, and you can kind of walk through a series of first degree relationships down to Cabernet Sauvignon and Cabernet Franc and Riesling, and they're all kind of related in a network of relatedness. And we see the same thing in apple here, and there's one kind of exception here up in the top right-hand corner is Honeycrisp. It only has 1 first degree relative in the collection. And Honeycrisp is an interesting example because what we're seeing here when we see this interrelatedness among cultivars, it does suggest that breeders have made use of the tried and true tested varieties that are successful in the market. It makes sense to go and use those as parents in the next generation. That's what generates a huge number of first degree relatives in the collection. And with Honeycrisp, they did, when they made the initial Honeycrisp cross, they thought that it was a cross between a Honeygold and Macoun, or Macoun, I'm not sure how you pronounce it. But, and it turned out with genotyping afterwards that neither of those were actually the real parents of Honeycrisp, that they're two other varieties. And so Honeycrisp is the only top successful cultivar here that is not interconnected to the other ones. And that shares very few first-degree relationships with other varieties in the USDA collection.
I suspect, Sean, that that's going to change quickly too, because Honeycrisp is used so much for breeding programs now, and so many of the new releases that are coming out of breeding programs use Honeycrisp as a parent. Yeah. And so I'm gonna just break in here because we do have a seminar that ends at 1:30, and want to allow for at least a couple questions. And I know that students have to move on for other classes. So maybe we can just leave your concluding slide up here and people can read through that, and just let people, either if somebody wants to raise a hand by using the little button below the participant pane, or just jump in here and ask Sean a question now.
Hi, I have a question.
Go ahead.
Can I ask, with such less genetic diversity, does that affect the nutritional value more because they're not bringing in new traits, or does that not have anything to do with it?
Yeah, it's funny that, you know, this less genetic diversity argument may not really apply. Just because things are related to each other doesn't necessarily mean that overall there's less genetic diversity. They haven't been inbreeding. It's not like we've been inbreeding varieties. They are relatively closely related to each other, but they're still extraordinarily diverse and heterozygous. Whether that results in less nutritional value, I think that just needs to be measured, right? I think that's a measurable thing. If anyone could actually agree on what nutritional value actually is and how to measure it, but that's outside of not my department.
Thank you.
Another question? So Sean, without another question right now, maybe if you just wanted to do any acknowledgements, and then maybe we can just hang out. I know that students may have to sign off, but we can hang out for a few minutes, and then you have another meeting. And I want to just, if everybody could give Sean a virtual acknowledgement and thank him for his time and sharing this data with us today. Really appreciate what you have done, and your research is really fantastic.
Right. Thank you very much for your time. I appreciate it.
And Sean, if you do have any acknowledgements, please share those.
Yeah,
now.
no, that's it. I did them all at the front and acknowledged everybody right up front. So this is it. I can direct people to the website and then to our Twitter feed. So if you'd like to learn more, then you can follow us on Twitter.
It's a lot of fun to follow Sean on Twitter, and he does, you know, of course, my interest is in the apples, but also in grapes and many other topics as well, including hemp now. And I see Larry's on the line, so he might want to follow Sean's Twitter feed. And there's also a very cool video on Sean's website.
Oh, sorry, I just thought of a question. Is it okay if I ask?
Go right ahead, Connor.
So one of the things I was wondering is I noticed that there were some cases where you were able to find loci that may be associated with adaptation, but it seemed like, as you said, there weren't, like, for not all the major traits, there weren't always the super, like, smoking gun, like, super obvious things. And I was wondering if you believe that for many of these traits that that adaptation could be polygenic, and if so, like, how do we actually, like, tell what genes are involved in that? Because I know that if adaptation is polygenic, you don't always detect that in these FST scans super easily.
Exactly, yeah. So if you have a single gene of major effect, imagine a Mendelian locus that gave a particular advantage, and it rose in frequency and showed a strong signature of selection, then that would be very easy to detect. But of course, if that trait that's been selected for is controlled by 1,000 genes, then you can imagine doing a genome scan, you're going to come up probably with nothing, depending on how much power you have in your analysis. And that's where we essentially lack power because we haven't saturated the genome with markers. We don't have really great phenotype data from, you know, multiple individuals and replicates and all the sorts of things that you can imagine having in a population of corn or rice or something. And so it does become more difficult to detect that kind of polygenic selection. The other thing to consider is that there may have been no selection, right? Haven't been really much, right? No, we don't know how much selection occurs in each of the crops that we're making use of. So, you know, in some cases there might not be much, in other cases there may have been intense selection for certain traits. And so without knowing what, you know, what proportion of the genome do you actually expect to carry a signature of selection a priori, you don't really have an a priori idea of how much should be under selection. So really what you're doing in these kinds of analyses is looking for the lowest hanging fruit. And when you do an analysis, for example, on the color, you find a really strong signature of selection for color. And so that makes a compelling story, and you've got strong evidence that, yep, color was selected for. But you're more likely to pick up color because it happens to be controlled by a one major effect gene, that MIB gene. And you may not find it for things maybe like firmness or for other loci controlled by, or other traits controlled by too many, they're too polygenic, and you won't find that signature of selection with the data that we currently have, or even with really great data sets. If it's controlled by enough genes, it might be really difficult to be able to detect. So yeah, you're absolutely right. The genetic architecture of the trait is critical for your power and ability to detect it in these sorts of genome scans.
Anna, did you still have a question?
No, I'm good, thank you.
Jonah, did you have a question?
Could I ask?
Yeah, I do have a question. Can I ask it real quick? My question was, over what time scale are you really looking at this ancestral selection? Because I think we all realize now with all of these breeders that obviously there is a lot more selection that we see on specific traits. So what is the time scale of that kind of ancient selection from the more less derived varieties?
Right. So when looking at, for example, the difference between the Domestica and the Seversii, you'd be looking all the way back to when they shared a common ancestor. And the same thing applies for each of the analyses. So when we're comparing red and green apples, what you're looking at is the differentiation since red and green apples diverged from each other, which might be a really long time ago. So it's not really, putting a time scale on these things is a little difficult. But it is, in fact, in some cases with really great data, you can do this in terms of counting up the number of generations and model it out. That might be a little difficult, I think, in this situation with a long-lived woody perennial that can, one of the things with apples is that, you know, you can mate with your you can have an apple mate with its great-great-grandparent because there's no real generational time, which makes the relatedness networks kind of strange, and the idea of time and generations a little strange as well, but because you can vegetatively propagate them for 200 years and it remains the same individual for 200 years. So might be tricky, it might be possible, but I'm not able to give real timelines for the timing of the selection for these different things. But needless to say, certainly that breeders have honed in a lot more over the last 50 years on certain traits than was the case before. And when I say breeding, you know, 500 years ago, it may not have really been all that intentional, and it may not have been, you know, making crosses and capturing pollen and making intentional pollinations. It may have been just keeping the offspring out of from random seedlings that looked the most desirable. So.
And Sean, somebody else, I think, had a question, and that'll be our last one because you'll need a couple minutes, and then you have a visit with the grad students starting at 1:40.
Great.
Did somebody have a question they wanted to ask?
Yes. Here, actually, Shushenk, and I have a quick question for you, Sean. And so you mentioned about the NAC18 and those SNPs, actually. So what's the nature of those SNPs? Is this loss of function or, you know,
So we're collecting GBS data, so we get whatever SNP happens to land nearby and is probably in LD with the functional allele. That's what we always assume. But that SNP actually happens to be a non-synonymous SNP in a binding domain of the NAC gene that's described in that plant genome paper. So it may be functional, but we've done some additional sequencing of NAC, and it turns out there's all sorts of variation in NAC that could be functional that is linked to that D5Y mutation. And we're going to do a more comprehensive sequencing of that entire gene across a large number of accessions, plug that genotype data back into the GWAS and see which of the markers is really most strongly associated with the phenotype. There's also like a huge deletion right in the middle of one of the domains too. So it could be a presence-absence variation, a copy number variation, or a structural variation going on within that gene. So we're going to try to tease that out with a little more additional sequencing. But to date, no, the causal allele at that locus is not known.
Cool. Thanks, Sean. And boy, you know, if it was up to me, I would just say this is the rest of our day and we'd just keep you on the line. But I know you have other appointments for the afternoon and other people have other activities. So again, everybody, thank you. Join me in thanking Sean for his time and his sharing his expertise and his research with us today.
Thank you.
Thanks, folks. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.